Assalamu alaikum, this is Snan and today we're going to talk about fat burning. Fats burning, aka fatty acid oxidation, or we also call it beta oxidation. It occurs in mitochondria in three steps. The first is activation, the second is carnitine shuttle, and the third one is beta oxidation. Now in the first step, as we know that fats are stored as triacylglyceride in adipose tissue. So these fats are acted upon by an enzyme called adipose triglyceride lipase, which converts it into diacylglycerol. Now this diacylglycerol is acted upon by an enzyme called hormone sensitive lipase and converted into monoacylglycerol. Now this monoacylglycerol is acted upon by an enzyme called monoacylglycerol lipase, which breaks it down into glycerol and three fatty acids. The glycerol goes to the liver and converted into glycerol 3-phosphate by phosphorylation, or this glycerol 3-phosphate can also be converted into dihydroxyacetyl phosphate. If it is G3P, then it is used in triacylglyceride formation in the liver, and in the form of DHAP it is used in gluconeogenesis. While talking back about the fatty acids, the three fatty acids in blood move to the blood. The red blood cells can't use it there because they don't have mitochondria. So these fatty acids bind to albumin, and the albumin takes them to the inside the target cell. Inside the target cell, the fatty acid is acted upon by an enzyme called fatty acyl CoA synthetase, which is an enzyme of the outer mitochondrial membrane. Now, in this process, 2 ATP are utilized, converted into ADP, and a CoA molecule is added to fatty acid to convert it into fatty acyl CoA molecule. This CoA molecule is also called the activated fatty acid. But a problem here arises that this fatty acyl CoA can cross the outer mitochondrial membrane, but it can cross the inner one, the inner mitochondrial membrane. So for this problem, we have a solution of the carnitine shuttle. In carnitine shuttle, the fatty acyl CoA can cross the outer membrane of the mitochondria, so it comes into the intermembranous space. Here, an enzyme called CAT1, full form is carnitine acyl transferase, acts upon it and separates the CoA group from it and adds carnitine with it. So it converts it into fatty acyl carnitine, removes CoA from the fatty acyl CoA and adds carnitine in its place, so converting into fatty acyl carnitine. Now this fatty acyl carnitine can cross the inner mitochondrial membrane through a translocase where it is acted upon by CAT2, that is carnitine acyl transferase 2. This carnitine acyl transferase 2 takes the CoA molecule, add it to the fatty acyl carnitine and removes the carnitine from it. So we got the initial fatty acyl CoA. While the carnitine produced in the matrix is transported out of the inner membrane via a translocase, which is used again. So this is the carnitine shuttle, and now fatty acyl CoA is inside the matrix of mitochondria where beta oxidation. Moving towards the beta oxidation. Now in beta oxidation, we are concerned with the alpha and beta carbon of the fatty acid. It occurs, as I have explained earlier, in the mitochondrial matrix, but there is a catch. It can take up to 22 carbon fatty acids. Above it, it cannot oxidize. So in what happens in beta oxidation, the two carbon fragments are successively removed from the fatty acyl CoA until there is a single two carbon compound is remaining. So beta oxidation occurs in four reactions. First one is the first oxidation, which produces FADH2 molecule. The second one is hydration. The third one is the second oxidation, which produces NADH2 molecule, NADH molecule. And the last one is thiolytic cleavage. Now, the beta oxidation is different for even number of fatty acids and different for odd numbers of fatty acids. Talking about the even number of fatty acids like palmitoyl CoA, which contain the 16 carbon atoms. The first, as we have explained, that it occurs in four reactions. So moving toward the first reaction, that is the first oxidation. So a fatty acyl CoA, this is the journal, this is the alpha carbon, and this is the beta carbon. We are concerned with both these carbons. So an enzyme called acyl CoA dehydrogenase acts upon the fatty acyl CoA inside the mitochondrial matrix and producing NADH molecule, producing FADH molecule. FADH2 molecule, and it removes the hydrogen atoms from the alpha and the beta carbon, converting it into enoyl CoA. Here we can see there are no hydrogen atom in the alpha and the beta carbon atoms. So it converts into enoyl CoA. Moving toward the second step, this enoyl CoA undergoes hydration. That is, an enzyme called enoyl CoA hydratase adds water into the enoyl CoA. So it adds a hydroxyl group at the beta carbon and a hydrogen atom at the alpha carbon. So it has a hydroxyl group at the beta carbon, so it is named beta hydroxy acyl CoA. Now this beta hydroxy acyl CoA moves toward the third reaction, which is the second oxidation. Now in second oxidation, the beta hydroxy acyl CoA is acted upon by its dehydrogenase. This process yields NADH2, which is an energy rich molecule. What happens in this process? The hydrogen is removed from the beta carbon atom, leaving the oxygen there. So we can say that removal of hydrogen is equal to oxidation. So we can say that the oxidation of the beta carbon has occurred. And this is the reason this process is called the beta oxidation. So the carbon forms double bond with the oxygen, oxidation occurs, and this compound gets converted into beta ketoacyl CoA. Now there is a fourth step called the thiolytic cleavage. Now in thiolytic cleavage, the beta ketoacyl CoA is acted upon by an enzyme called beta ketothiolase. The beta ketothiolase cleaves the compound close to the alpha and the beta carbon and yields one acetyl CoA molecule, while it also uses another CoA molecule to attach it with the fatty acyl CoA. So one 2 carbon compound acetyl CoA is released and the remaining carbon fatty acyl CoA are present. So these remaining carbon fatty acyl CoA are repeated again and again into the cycle until we get the final 2 carbon acetyl CoA. Now the number of repetition of a fatty acid degradation is equal to n by 2 minus 1, whereas n is the number of carbon of the fatty acid. For example, for one beta oxidation, we have seen that in the first oxidation FADH2 is produced, which is equal to 2 ATP. In the second oxidation, one NADH2 is produced, which is equal to 3 ATP, while in the thiolytic cleavage, the acetyl-CoA is produced, which moves to the citric acid cycle and produces 12 ATP. So, in case of palmitoyl-CoA, the number of carbon atom are 16, placing 16 in place of N, divided by 2 minus 1. So the cycle will repeat 7 times, resultantly providing us 7 FADH2, 7 NADH, and 7 acetyl-CoA. But there is a catch. In the final step, where 4 carbon atom is remaining, its breakdown will produce 2 acetyl-CoA molecule. So in the final step of the thiolytic cleavage, there is an extra acetyl molecule production. So we will add another acetyl-CoA, and all of this add up to form 131 ATP on the degradation of 16 carbon palmitoyl-CoA. That's a lot of the ATP. But we have utilized 2 ATPs in the activation of the fatty acid, that is its conversion into fatty acyl-CoA. So a net of 129 ATP is produced by the beta oxidation of 16 carbon fatty acid. Still a good deal, I think. Now there is a question, what will the reaction for the odd number of fatty acids? For odd number of fatty acid, let us say 13 carbon fatty acid, it will oxidize up to 3 carbon fatty acids, and 3 carbon fatty acid is known as propionyl-CoA. Now this 3 carbon fatty acid is acted upon and called the propionyl-CoA carboxylase, which adds a carbon dioxide, a carbon atom into it, converting it into methyl mannoline-CoA. Here's the carbon dioxide. Now the propionyl-CoA requires 3 things. The mnemonic for it is A, B, C. A for ATP, B for biotin, and C for carbon dioxide, which is add to make a 4 carbon methyl mannoline-CoA. Now this methyl mannoline-CoA is in its D form, which is converted to its L form by an enzyme methyl malonyl-CoA racemase. Now its L form is acted upon by an enzyme methyl malonyl-CoA mutase, and this is the enzyme which requires the vitamin B12. Now fun fact, this is the one of the 2 reactions requiring vitamin B12 in the body. Now the first one,
नहीं मुझे मजाक नहीं उड़वाना अपनी चीज का, मैं नहीं बताऊंगा.
Now succinyl-CoA is produced. This succinyl-CoA can be used in the TCA cycle, also known as Krebs cycle, or can be used in the formation of heme. Now moving further, now we have said that the beta oxidation has a limit, and that is 22 carbon atoms. What happens if there is a fatty acid longer than 22 carbon? The answer is peroxisome. The peroxisome takes those fatty acids and cut them to 22 carbon shorter chains and then send them to mitochondria. Moving to the fun facts, which is a new addition to our lectures, a question arises, is there an oxidation of fatty acid other than beta oxidation? The answer is yes. There is oxidation known as alpha oxidation or gamma oxidation, omega oxidation. Now talking about the alpha oxidation, it occurs for branch chain phytanic acid, which is a product of chlorophyll metabolism. Now a catch here, that why this alpha oxidation of this fatty acid occurs is that its oxidation by acyl-CoA dehydrogenase cannot occur because this enzyme is not suitable for it. In place, an enzyme called the palmitoyl-CoA alpha hydroxylase acts upon it and oxidizes its alpha carbon instead of the beta carbon. So it is the alpha oxidation. Now our question for our viewers is, if there is a omega oxidation, then what and where it happens and in what condition, the winner will be given a special prize. Thank you.
Yep.